All right, and our final section, which is going to be 21.5 genetic engineering as a whole. When we look at genetic engineering, what we are discussing here is really anytime we are taking the DNA that codes for the production of a particular protein in a cell and how we are going to ultimately extract that DNA from a human cell and combine it with the DNA from, let's say, a bacteria cell. Now, this comes into play when we are creating insulin, as well as crop plants that have been genetically modified to either be resistant to herbicides or insects. They also can be genetically modified um, to to have different vitamins input into those. Let's say this rice here, it can be fortified with vitamin A, a whole lot more vitamin A than normal rice. So with looking at genetic engineering, let's focus on the crop aspect of this first. As someone who has graduated from Mississippi State and worked in the agriculture and forestry department for a period of time, I have worked with so many different professors and research specialists at Mississippi State University that are working in this area to ultimately make seeds for crops that are drought resistant, that are resistant to herbicides, that are resistant to different types of organisms that threaten to wipe out certain types of crops. And then also just to create seeds and different types of crops that can be fortified with a greater amount of vitamins. This is so vital, let's say in your third world country where food insecurity or really just malnutrition is something that individuals face on a regular basis. So to me, genetically modified organisms and genetically modified crops are something that is such a blessing and such a great source for the quality of human life in certain parts of the world. So I am a proponent of genetically modified crops. Now we do have to look at genetically modified crops because there are some people who argue that that it is not a good thing. So there are individuals who feel like because we genetically modify crops that there could be a risk of it not being something that is natural to our bodies. However, I do want us to go back and think about all organisms house DNA. DNA is made of 4 base pairs that are the same over the entire scope of living organisms. So adenine, thymine, guanine, cytosine, we share those same base pairs with a dandelion as we do with my child that's sleeping upstairs, as you do with the ear of corn that's growing in the farmer's backyard. All of those are based off the same genetically unique base pairs of DNA. So to me, I know individuals have concerns that GMO foods and genetically modified crops can be dangerous because really I think we just need to think about it in terms of we are just taking what we know of DNA and we are combining it to ultimately fulfill purposes that help us and don't harm us. So I'm a proponent. You might have a different opinion, and I'd love to hear about that. You can actually dive into that in the assignment that you'll start on here soon. But with genetically modified crops, if you're a proponent or not of those, we do need to talk about human insulin and how it is created, which I'm going to scroll down to this image right here. So let's see, I think, this is a depiction of how insulin is created. To create insulin, first, what scientists have to do is take human cells and extract actual human insulin from those cells, and then we must take the bacterial cell, which has its own DNA inside of it. Now, to try to make this image look a little less confusing, because I look at it and kind of get lost in it as well. If you're looking at it going, what in the world is going on? What you're going to want to focus on is we take a portion and this would be indicated by this pink portion right here. We take a portion of human DNA that contains the insulin gene from our human bodies. We take the portion of the bacteria cell's DNA, so their DNA right here, it's called the plasmid from the bacterial cell. We ultimately open up that bacterial cell's plasmid through a unique process of clipping those open to expose what's called sticky ends. Okay, so they're cut open in this open form, and then we can take the human DNA containing the insulin gene, and then we can form it with the bacteria cell and its opening, and then they can recombine, and we can ultimately stick that back into this bacteria cell. Now, what's unique about this is we have taken the human gene, we have put it back together with a bacterial cell. This bacterial cell will now begin the cloning process, which is just cell division, through however this bacteria cell ultimately reproduces, which is through asexual reproduction. So it will begin to replicate. So this one bacteria cell with the human insulin gene will now begin to replicate itself. So its one cell forms two cells. Those two cells form two cells. Those cells form two cells. And you can see that from combining our DNA with this bacteria cell's DNA, we have now formed a unique microorganism through biotechnology to completely act as a means of creating human insulin. So this is what someone who suffers with diabetes has to have to survive. Because if you think back to our unit on homeostasis, this is where you have to have insulin to be able to process all the sugar and the glucose that you're putting into your body. And if you don't do that, then you must have synthesized insulin being placed into your body. Well, this insulin is created through this process of taking a bacteria cell, merging it with a human component of this cell, and then allowing that bacteria cell, once it has the human components inside of it, to replicate and reproduce. Ultimately, through that fermenter and that fermentation process, we can gather the end result of having human insulin produced. It's amazing, y'all. I don't know if this kind of blows your mind, but it blows mine. And so this is pretty much it for the unit. I hope you enjoyed learning about some cool biotechnology.